talk a little bit about our case for higher oil peanut lines. We'll talk about how peanut breeding works at the University of Georgia, some of the genetic resources that are available to us, our strategy for moving forward from this point, and some goals and timelines. So peanut breeding is a very slow process, and so I just want to kind of share with you, how long it's going to take to get to the point where we have some really high oil varieties that are ready for the market. So I think a lot of people have probably seen this infographic from Bob Parker recently, just showing the price differential between what we're currently getting out of a typical ton of edible peanuts versus if we went to a straight crushing plan. All right, and so you can see that there's a $157 reduction in value per ton. But when it comes to peanut oil, it's shown about midway in this infographic here. We have just as good an oil product as most of the other vegetable oils. We have a fairly low saturated fat, nice high oleic concentration and high linoleic acid concentration. When you compare it to soybean oil, it's actually quite similar. It's more healthy than cottonseed oil and some of the others, and a very competitive product. When we look at prices, though, olive oil really commands the market for vegetable oils. Peanut is shown there in the kind of brownish gray. All right, when we compare peanut to other row crop type vegetable oils, it has a really good value point. And I'll let others who deal in economics tell you a little bit more about that. But when it comes to breeding and agriculture, how do we trim $157 of production costs off of that ton, okay, to make this viable? For a breeder, we think in pounds per acre. So for me, the easiest thing to do is just to make sure we produce another $300, $400 per acre worth of oil. We don't really want to decrease our inputs too much because that could decrease our yields. We want to maintain high yields so that we get high oil yields. And like I said, we need both high oil and high pounds per acre. So we also need high plant vigor. Growers, when they put a seed in the ground, they need it to come up quickly and cover that row rapidly so that they can conserve water and reduce herbicide costs and things of that nature. We also need good disease resistance, right? Growing peanuts in the lower southeast, we've got to have good TSWV resistance. We've got to have acceptable early leaf spot, late leaf spot, and white mold resistance. These are must-haves. Oil comes from the seed, so we want a high shell-out percentage, high meat percentage, and then good stable yield performance over dry land as well as irrigated acres. Another way we can save on costs is smaller seed. Plant more acres with the same number of dollars. And then, an idea that's been proposed is maybe these high oil peanuts could have a different seed coat, and we'll see some pictures later, see how that might be possible. Okay, so peanut breeding at UGA. So I want to share with you how we're going to get to the point where we have some viable high oil peanut varieties. And so to do that, I want to share with you some pretty simple examples of plant genetics. Okay, in this example, we've got a purple pea flower crossed to a white pea flower. The hybrid is going to be purple in this example. And then in the F2 generation, 1 out of 4 of these progeny are going to be that desirable white pea flower that we're shooting for. Okay, when we expand this to a 2-gene model, in this case, 1 out of 16 of those progeny are going to be whatever it is our desirable phenotype is, okay, that it will actually breed true into the next generation. Okay, so we need high oil, potentially small seed. We need high yield, TSWV resistance, white mold resistance, early leaf spot resistance, late leaf spot, high meat percent, and high vigor, okay? In a really simple example, that's 9 genes, okay? Really, it's way more than that. But just for the sake of example, that means 1 out of 262,000 individual plants in this particular breeding population are going to have all those traits stacked within one genotype. And I've got a bit of a typo here, but basically, if it's an all dominantly inherited trait, 7.5% of the population is going to have what we want. And if it's all recessively inherited, it's that really long fraction of a percentage. So somewhere between close to 0% to 7.5% is what we're looking at when we start this breeding process. There's a lot of ways that we can kind of stack the deck in our favor. Let me just show you how breeding typically works within the program. So we'll take two varieties that have, the list of characteristics that we want. We want to start out with mostly good by good crosses so that, the odds are more in our favor. And we'll take those hybrids. In the first year, we plant those in the field. We make selections among those F1 hybrids, and the following year, we plant entire populations to the resulting F2 population there. We dig those peanuts and make visual selections in the field. In this example, we're selecting 4 individual plants from hundreds, if not thousands, okay, that really stand out and look to have the combination of traits that we're looking for. And so during the off season, we'll take those individual plant samples, we'll shell them out, we'll measure everything we can possibly measure on those individual plants, and then we'll plant entire rows from each of those individual plants. So we've got 4 what we call F2 derived F3 progeny rows, okay? And then we repeat the process, okay? We're constantly evaluating for the disease resistance packages we need, constantly evaluating for yield potential and other quality factors. So in this case, we've selected 4 more plants, we shell them out, make further selections, and then plant more progeny rows the next year. We make some more plant selections, and then at that point, we consider these fixed lines. So these are fairly homozygous, they're homogeneous, the seeds are gonna breed true, we can feel confident that they're gonna be uniform from year to year. Okay, so in the F5 generation, we increase our seed supply, and then plant F6 preliminary yield trials. So in these are replicated yield trials where we're comparing these new breeding lines to the commercial checks that are available on the market. Okay, if they make all these cuts, then by the 7th year, we go to the F7, F9 replicated yield trials across Georgia especially, sometimes into other states. And if after 10 years they've made all the cuts and they're still looking good, then we release them as varieties and they go on to foundation seed increase and the various crop improvement associations for each state. All right, so this is a picture of our crossing block in the winter. We do all our crosses in the wintertime. That's when we have time to make crosses. Usually we actually start crossing while still harvesting in the field. But anyway, this is our crossing block. And so the afternoon before cross, we'll go and we'll find some flower buds and we emasculate them. We remove all the male parts and petals that we don't need. And so what you see here, this hair-like structure there, that's the pistil, okay? And so after we emasculate the flower and get it ready for pollination, we put this little string on there, and that'll help us identify the flowers that we need to pollinate the following morning. That following morning, we pull our male flowers from whatever variety it is that we want to cross-pollinate with. We get a little bit of that pollen on the tweezers and dust it on that pistil you just saw. This is what a successfully pollinated flower looks like. Again, it's got a little string to help us identify the pollinations. We cover it back up, and then we watch that cross for 4 to 10 days looking for a successfully hybridized peg. So you can see the beginnings of a peg here. The flower is still attached with that string, showing that it's a hybridized peg. So then we need to mark those to make sure that we can harvest those hybrid peanuts. And we'll grow them out until they reach maturity and harvest them. And the peanut will still have the wire tag around it. We bag them up, dry them down, and then carefully hand shell every single peanut that we cross. These are some of the most expensive peanuts you'll ever come across. A lot of man hours goes into every single one of these. So the picture on the left here shows an F1 breeding nursery. So those are the F1 hybrids. We usually hand plant those because there's not a lot of seed. And then we'll harvest those, and the following year we plant our F2 breeding nurseries into a really big field. We want plant as many of these F2s as we possibly can so we can select those really high-performing individuals that stand out. This is what that looks like. So you can see the plant on the left is lower yielding than the plant on the right. It's probably hard to see in this picture, but those pods are eaten up with TSWV on the left, and the pods on the right are very clean, look really nice. We'll select thousands of those every year and then individually pick them using these individual plant pickers. It's basically just an expanded metal grate over a bucket. We save all those seeds, shell them out, make selections during the off season, and then we'll plant these progeny rows, like I said earlier, in the field. So the progeny row on the left there, you can see it's got a lot of TSWV. The progeny row on the right also has a lot of TSWV. You can see some fairly resistant plants segregating, so that may be a cross between a resistant parent and a susceptible parent. And then the progeny row in the middle there looks very resistant to TSWV. So we're selecting not just progeny rows, but also individual plants within those progeny rows that have the mix of characteristics that we want. Once we have a fixed line, we use these self-propelled combines to harvest pure seed. And then they go into these replicated yield trials. And we'll plant these all over the state. This is what our yield trial plot picker looks like. It's a 2-row KMC fixed with a bagging attachment on the side. Okay, so genetic resources. There's a whole lot of wild species genetic resources with very high oil content in excess of 60%. The problem with the wild species is it takes a really long time to introgress those really valuable traits into something that a grower would be willing to plant. You know, it's hard to get rid of some of these wild characteristics that aren't suitable for production ag. But fortunately, we've got a lot of colleagues that work with wild species that can help us with some of that. Peanuts are also grown in China and India as primarily an oilseed crop. And so there's a lot of really good genetic diversity out there for a cultivated type peanut with high oil content. This is just an example of some data in a cross between a low oil and a high oil type peanut. These are cultivated types. So, the range is between 42 and 56 here in this environment, or 48 and 60%. So we can get some really high oil contents in cultivated peanuts. This graph down here, these same researchers were trying to show the comparability between environments for oil content. And so, if we're growing high oil peanuts in South Georgia, those will also be high oil peanuts in Alabama. There is some environmental influence on percentages, but we don't really know, what kind of effect that is, other than, the performance will still be comparable across environments. So as far as genetic resources, we also have genetic tools that are available to us today that weren't available a long time ago. This is an image of what we call electrophoresis gel. These are what we call SSR markers. When I was in grad school, this is what we were using. Okay, we could only screen a couple hundred markers on a population, and that would take ages to get it all done. This is from a study just a few years ago using a technique called genotyping by sequencing, where we use thousands of markers to help us map the location of really important genes. Okay, so we're able to do just leaps and bounds from what we were doing just a few years ago. And then today, we can leverage tens of thousands of markers to figure out the location of the gene. So this is a project that we're working on right now, looking at a gene for seed size, and we've really nailed down the location of that gene very precisely with this tens of thousands of markers in this population. So when I say mapping a gene, what we're doing is we're identifying the parts of the chromosome that influence the trait of interest. So this is from a study from China, I believe, where they were mapping the genes for oil. And so they had really good results, just indicating that mapping these genes will really help us in our breeding effort. And they've already shown some success. I'm confident we can also. This is just showing the relative effect of those genes when pyramided within genotypes. Okay, so we've got good genetic resources. I've shown you kind of how the peanut breeding program works at UGA. And so now I want to share with you how we're going to go into this new breeding effort and what our strategy is. So first of all, we need genetic diversity. So within the runner peanut varieties that we currently work with, our max oil percentage is around 50%, okay? And there's not a whole lot of variation around that. You know, it may be as low as 48 to as high as 52. So fortunately, within the U.S., we have a really amazing national plant germplasm collection system. There's one in Griffin, Georgia, that's curated by this gentleman here, Dr. Sham Tollery, and he curates the Vigna as well as peanut germplasm collection. So just before planting season, I requested a whole lot of high oil germplasm from him. Now these are lines that aren't adapted to our region, so currently I'm evaluating them to see if they can be used as parents within a high oil breeding program. You know, I showed you that slide where, we're trying to get a lot of genes into one single genotype, right? The odds of having all those in one is pretty low. So I want to make sure that we're selecting the best possible parents with the best possible yield and also high oil. And so that's what we're doing in the field this year. Some of these are some Valencia types. Some of them are highly susceptible to TSWV, which is kind of a non-starter for us, but they have some really high oil content. So you can see this plant on the left is just eaten up with TSWV. These other plants on the right the same way. You can see some really interesting seed coat colors. We've got dark brown and a red, which when we talk about non-edible peanuts, this may be a really nice solution to help us with segregation. You know, when you pop open a load of non-edible type peanuts and you see that brown seed coat, it would make it very clear. So some of the parents actually look pretty good. So this is a typical runner growth habit. Looks to be fairly resistant to TSWV, and hopefully this season we'll evaluate its oil content to see, just to confirm what the literature says about the oil content for this particular line. And there's some other options too. We've got some really nice, they're not cultivar ready lines, but they might make good parents. Okay, and so we're gonna enter these guys into our crossing block in just a couple weeks. We'll start planting them, and we'll make a lot of crosses. And depending on how the selections go this fall and how these oil contents go, we may drop some of those crosses, or we may promote some of them for accelerated development. So like I said, we'll enter them into our crossing block, and we'll kind of wait to see what the data says before we kill some of these. So a big part of our breeding strategy is going to be, utilizing technology in our favor. There's a whole lot of really neat technology available to plant breeders these days. This is one of those. It's a high throughput seed sorter. Some of you, especially those in the shelling industry or manufacturing industry, may know this. It's called a Q sorter from Qualisense, and it's capable of sorting high oleic peanuts from normal oleic peanuts at 20 seeds per second. It's really, really fast. And so we're using it in our breeding effort to help us in the development of both normal oleic peanuts as well as high oleic peanuts. But the way it works is it has a near infrared camera right here and a regular visual spectrum camera right there. And in this diagram, you can see the hopper for the peanuts is on the left, and it uses a vacuum belt, similar to like a vacuum planner or air planner, where you've got holes in the belt and the vacuum is applied behind it and the seeds stay stuck to the holes. It passes through the machine. It goes through this visual spectrum camera where it takes a lot of measurements on the seeds. It comes across this near infrared camera where it evaluates the spectrum reflectance of near infrared light, which tells it whether or not it has a high percentage of oleic acid or a high percentage of or a low percentage of oleic acid. And then, once the computer makes that decision, it'll shoot the high oleic seeds into bin 1 or the normal oleic seeds into bin 2. And if for some reason it's a damaged seed or it didn't see the seed very well, it'll put that in the third bin. This is what a typical data set looks like when we're looking at just oleic acid. And so we've got a whole bunch of, these are individual plant samples. Every single one of these dots on the graph represents an individual seed. Okay, and so the blue dots represent the first pass through the machine, and the red dots represent the second pass through the machine. And it's basically confirming that these are high oleic samples, and then it's getting rid of anything that's slightly, under the 70% threshold that we use. And basically, in these high oleics, that's immature seeds. And then over here on the right, we've got the normal oleic seed samples that I use as a check. And then we've got some heterozygous plants. So these are plants that have one copy of the high oleic gene and one copy of the normal oleic gene. And so their seeds are segregating. So they get 25% high oleic seeds and 75% normal oleic seeds. And the machine is capable of separating those out. And so it's been a really big help for our breeding program, maintaining genetic purity and keeping the two programs, separate from each other. This is just showing the part that gets selected after the second pass through the machine. This is what the samples look like. So you've got, again, 75% normal oleic seeds and 25% high oleic seeds. We put those two different samples in bags. So the high oleic goes in the Ziploc bag and the normal oleic seeds go in the brown paper bag. And then we'll use the high oleic seeds to plant in our high oleic nursery and the normal oleic seeds to plant in our normal oleic nursery. Whoops. So we intend to use this thing for oil too. So we just purchased the calibration for it to basically do the same thing it's currently doing on oleic acid, but this time for oil, so that we can breed for oil in a more high throughput manner. And essentially, the breeding program is going to look very much the same. We'll make plant selections in the field, we'll shell them out, and then the difference here is that we're going to be using the Q sorter at every step of the process to increase oil percentage. So, assuming we take a conservative approach, we're looking at about 10 years to develop a dedicated high oil breeding line that's well adapted to the southeast, that has all the list of characteristics that we want in a southeastern runner cultivar. So, like I said, breeding program is going to look largely the same, except now we're selecting for oil in addition to all these other things. High vigor, good adaptation, broad performance, good disease resistance, high shell out, and potentially smaller seed. So this is what the timeline looks like for developing a new variety. This year we're making the crosses, and each field season we'll be evaluating these populations on through to elite yield trials in 2032 before we release a variety. There's a lot of things we can do to accelerate that, to shave a few years off this process. One of the things that you lose, though, by, say, growing seed out in a winter nursery or in a greenhouse over the winter is you lose an opportunity for selection. You guys know that not every season is the same, right? We have lots of rain some years. Some years we may have very little rain. We may have hotter TSWV pressure some years than others. And so, especially whenever you're breeding with new sources of genetics, it's very important to take your time and make sure you evaluate these breeding populations for as long as possible, so that whenever we hand over a variety to you as the grower, you know what you're going to get, right? We don't want you to lose money by investing in one of our varieties. So goal number one is to develop germplasm that has high oil, that's adapted to the southeastern U.S. This is stuff that we can share with other breeding programs like the University of Florida or Auburn. Also, goal two is to map the genes that are influencing oil percentage so that we can make breeding for high oil content easier and more economical. And then eventually, hopefully, develop a southeastern runner variety with high oil. For some people, this may look like a daunting timeline. For me, that's just plant breeding. 10 years it's not a lot of time when you when you're a plant breeder, that's just the way it is. But we do have some plans to try to use some winter nurseries and things so we can try to accelerate the process. I want to thank Dr. Bill Branch, who I work with closely there at University of Georgia, for his mentorship and just willingness to give me lots of really good advice. He's been the peanut breeder there at UGA for over 40 years now, and I think we all realize that we're really lucky to have that gentleman as a plant breeder in our state. I want to thank my little ragtag crew here, Shell, Jesse, Drew, and Ethan. Ethan's gone on to bigger and better things. We've got a whole new crop of student workers coming in, so really grateful for the work they do in our breeding program, and I definitely couldn't be here without their help. I want to thank the help of Dr. Sean Tollery sharing germplasm from his collection there, from the USDA collection, and Don Kaler and the Georgia Peanut Commission as a whole for their leadership. They've encouraged me to start working on this, and I think it's a really good idea. Again, I'm just really grateful for everything that they do for our research program and also for our growers. Without that, also, I should say I'm really grateful for the funding to help support our breeding program from these other agencies like the Peanut Research Foundation, Georgia Seed Development, University of Georgia Research Foundation, National Peanut Board, and Georgia Crop Improvement Association. Okay, thank you.